welcome back to our Saturday edition of the Big Health Data Science Center Conference. I'm so honored to be able to introduce our next speaker, Dr. Holly Gaff. She's a professor of biology at Old Dominion University. She received her Ph.D. in mathematics from the University of Tennessee. She is truly an eminent expert on mathematical modeling, infectious disease forecasting, with an emphasis on tick-borne diseases. Throughout her career, she's maintained an impressive extramural grant-funded portfolio, including grants from NIH, CDC, and a variety of other sources. She's also amassed some really incredible honors for her teaching, her service, and mentorship of the next generation of mathematical scientists. She is a one-stop do-it-all. She's really an impressive woman. And today she's going to talk about spatial ecoepidemiology of tick-borne rickettsial pathogens. And just a quick housekeeping note, if you would please mute all of your microphones. I'm gonna do the same, and then place any questions you have in the chat and we'll get to it at the end. Thank you.
Excellent. Thanks, Melissa, and thanks for the invitation to to give a talk. I'm always happy to have an audience to talk about modeling and ticks and a few other things. And so hopefully a lot of data that we've collected over the years and what we can do with it and where we're going from this. So hopefully this will be some part of this talk will be of interest to everyone here. I'm happy to answer any questions that you have at the end. So please write those down as you think of them so that you don't forget them, because if you're like me, you'll be like, that was a great question, but I don't remember what it was. So again, I'm going to talk hopefully some of the stuff you may know, but hopefully you'll learn a few things, and then I look forward to some conversation. So just a little bit about ticks. I know everyone does not work on ticks, even though they're amazing little creatures, and there's a lot of work that needs to be done in this area, but I wanted to just give you a little bit of the biology background so that you have a little better understanding of the systems that I'm working in and the data that I'm collecting and what I'm doing with it. So some fun facts that you can now fascinate your friends with is that ticks are worldwide. They are about 865 species around the world. They're on all continents, including Antarctica. They usually live in the nests of animals down there, but you can't go anywhere that you won't find these little pests. And of course, as you probably are aware, they are vectors of human pathogens in the United States. They are the #1 vector of human pathogens. So even though we spend a lot more money in research on mosquito-borne diseases, tick-borne diseases are of a major concern. They also will vector wildlife and veterinarian pathogens. And so they're kind of this vector that causes a lot of problems. They are divided into a couple different classes. There's hard ticks and soft ticks. We're going to talk mostly about hard ticks today, but there are another class of ticks called soft ticks, which have a slightly different life history that I've also worked on for other things. But today we're going to focus on the hard ticks, which are probably the ones you're most familiar with. They are actually not insects, nor are they spiders, but they're a special subclass of acarids. And so they're kind of this interesting invertebrate that has its own little life history that's really unique. I mean, they're kind of amazing in all the things that they do. You know, for example, they have 6 legs for some of their life stages and 8 legs in other life stages, and they have metamorphosis between these life stages. And so it's pretty interesting. Speaking of their life history, just to make sure that, again, the terms I'm going to be using for how they evolve through their life. Ticks have what I call a punctuated life history, and this is kind of the interesting dynamics part of it. So the lifetime of a tick, they start as an egg, and then as they emerge from that, they hatch, they harden, they become larvae. They'll come up, they'll look for a blood meal. If they're successful in that blood meal, they'll fall off usually. Molt into a nymph, they come up again, they look for a host, they'll feed if they're lucky and get their blood meal, then they'll fall off, they'll become molt to be an adult, then they'll come up and feed one more time, mate, the female lays eggs, they both die, and they start the process again. And the part of this process that I find most interesting from a data management and thinking about modeling perspective on the mathematics side is that this process takes years. And so the interaction time between vectors and hosts is often the part that we think about from a dynamics perspective as being the most important time. But in a lot of ways for ticks, they have an equal, if not more important, survival off host in the environment time, which is an interesting way of trying to really understand what's going on. during the life of a tick. And so understanding what's going to be predicted for the next life history. Soft ticks, by the way, actually live a lot longer than hard ticks. They had a paper or announcement a couple weeks ago that we now have a 27-year-old soft tick that has been around for that long. So these ticks, I mean, they're amazing in how long they live, and how poorly studied and how little we know about this life cycle. The ticks themselves will actually feed on almost every vertebrate out there has some tick that will feed on it. Some ticks are very picky and will only feed on one host specifically. For example, there are ticks that will only feed on rhinoceros, and so are either extinct or endangered of going extinct with their particular host. And others are very widespread in what they'll feed on, and they'll feed on pretty much anything that's moving. And every survival technique between the two. So ticks just, some will chase you down, some will just sit and wait. They each have their own unique biology, their unique life history, but overall they are very hardy and widespread. We've tried all different ways of killing them, and they can survive for very long times in very harsh conditions that just amaze you. As a reminder, again, the reason that I'm most interested in doing a lot of this work is, of course, because it's associated with disease. While it is a fascinating system ecologically and mathematically, my motivation is to really understand how we can help prevent future tick-borne diseases. And so for humans, of course, this involves a lot of Lyme disease, which has somewhere it's reported diseases of about 30,000 new cases every year, but the estimate is probably closer to 300,000 cases would be the more accurate estimate. Spotted fever group rickettsiosis is a real problem here in the Southeast. So that's Rocky Mountain spotted fever is the one you've probably most likely heard of, but there's a number of other related rickettsias that can give you spotted fever outbreaks of different kinds. If you travel internationally, tick-borne encephalitis is a major problem throughout much of Europe and over and through Russia. For animals, I work a lot in Africa as well, and there's a lot of challenges with animal-based diseases. So a lot of livestock are affected by East Coast fever or heartwater. And in Africa, that's a real economic challenge since their economy is often built on their livestock. So here I am at Old Dominion, and as Melissa said, my background is actually in mathematics. And so I worked on tick-borne diseases since grad school, and I often, I got interested in them because I was interested in math, I was interested in public health, and my PhD advisor happened to work on a committee of somebody who was working on an outbreak of tick-borne disease in Cumberland County, Tennessee, and thought it'd be interesting to build a model for that. And so I did, but I made a model. I looked at some exploration of some of the dynamics of this interesting system, but I was very frustrated that I didn't have good data to support the model. I continued working on these models for the next 20 or so years, not really happy with a lot of them, but at least exploring the potential dynamics of what the system was as best I understood it. And the challenge with tick data was, like a lot of health data, we tend to only collect it when there's an outbreak. So like I mentioned, I got that data from an outbreak in ehrlichiosis. But what we didn't have was the data from when there's not an outbreak. So knowing what is going on when there's no outbreak going on is as important as knowing what's going on when there is an outbreak going on, to know which pieces are different and what's actually driving the dynamics of the outbreak. So I got here at Old Dominion University in Norfolk, Virginia. And at Old Dominion, there was an internationally recognized tick expert, Daniel Sonnenschein. And so I got here and I went to Dan and I said, I need these data to make my models better because they're terrible. And he looked at me and he said, well, no one's collected data thinking about modeling. You need to go outside and just collect a few data points, and then you can come back inside and build your models. And so we joke about this now because he did indeed trick me into going outside as a mathematician, but up to that point, I had sworn I would never do field work. And so this was 2007 when I got to Old Dominion, and there I was going out now doing field work. And so we started what's now become this long-term tick surveillance project here in the Hampton Roads area of Virginia. We go out, we collect ticks from the field, we bring them back. I have collaborators who do amazing lab work and do the PCR and the sequencing and do all the dynamics of testing, that they can be able to give us the understanding of what populations of ticks we have, what the pathogens are carrying. And then we take all this data and come back and do some mathematical modeling to really understand where the high risks are, what's driving the risks, and what the possible control options are. So just a little bit about the data that we've collected over the years. So I have collection sites throughout all of Virginia. So right here, I'm in Virginia, I'm in the southeast corner. All of these orange dots are some long-term field sites. And so we started this project in 2009 And so each of these spots, we have gone out and have multiple habitat types within each location. And then we flag for ticks at each location at least monthly since 2009. And so what we collect ticks, right? If you aren't familiar with collecting ticks, we do mostly what's called flagging. We take a 1 meter square white denim cloth, attach it to a dowel rod, drag it through the vegetation, put the ticks in vials and bring them back to the lab. And that's the majority of the ticks that we collect. But of course, not every tick is looking for a human or something large to bite. And if they're not looking for something large to bite, they're not likely to be in the part of the vegetation where a flag would collect them. So we also do trapping and other animal collection so that we can get the ticks off the animals. So we do rodent live trapping. We go to hunt check stations and work with hunters to get the ticks off their deer. We stop at roadkill, which is always a fun and sexy thing to do. We work with veterinarians. We have some humans that actually give us data off themselves, so the mosquito control guys and some of the wetland mitigation folks from the Army Corps. We've done some mist netting. We have ticks from reptiles. And again, our major goal was to really understand the entire system, not just small pieces of it or during outbreaks. And so the data from flagging alone, this is just our flagging data normalized by effort, which has changed a little bit over the years just for weather related issues or hurricanes, has varied some by year. We've collected about 250,000 ticks at this point, and we've processed them. We have not tested all of them, but we're working on it. And what you can see is some years we get lots of ticks. So 2012 was a really particularly high tick year with over 45,000 ticks that year. And as I was going through my data, I was starting to look for patterns and trying to understand what was going on. And, you have all this data sitting around and you think, well, that would tell me that there's something in that year that made it really good. And for many years after that, my hypothesis was that year we actually didn't go below freezing the entire winter of 2012. And so I was like, well, look, what warm winters will make more ticks. And fast forward, here I am, 2018, we go back, we're almost at the same level of 45,000 ticks. And that winter was actually one of the coldest winters we've ever had on record. And so that shot down that hypothesis. So now I'm back to the head scratching part of not really knowing exactly what's driving these fluctuations in populations. And now I have a new head scratcher here in 2020 where we still went out, even though it was COVID and limited to just my grad students and I without the usual crew. We still managed to go out and collect all of our ticks, but there were no ticks to be found. It was just an incredibly quiet year. So we joke the ticks were also hiding at home, or my current hypothesis is that animals were more active and so therefore there were more ticks being picked up. But who knows? We're waiting to see what happens as the numbers come back. This year, certainly 2021 certainly looked a lot more like the previous years, but we've also had hurricanes and blizzards and no blizzards. And so we need hopefully another 10 years of data. Maybe we'll have some idea of what's going on, but it's been an interesting set of fluctuations. And so I'm starting to use this to build the models I'm going to talk about. One thing I'll point out is this is total ticks that we've collected. And one of them, if you've lived in the Southeast, the majority of ticks that are down here are the Lone Star tick. So if I take out the one particular dominant tick species and look at my data, I get a completely different pattern. And so while these ticks are living in the same habitat with the same hosts available to them, we get totally different dynamic patterns. And so 2021 was actually the highest year of non-Lone Star ticks that I collected. And so we could make graphs for each of these different species and look to see what years they're high and low. And it's just amazing how each species has its own dynamic pattern from year to year and within the years. To be able to help us understand what's going on is really a challenge, because each tick seems to have its own preference for habitats. And then there's host dynamics on top of this over these years, and it's just a lot to still work on. And so this is, of course, oh, by the way, this is my public health moment. So from our data here in Virginia, we always like to make sure people are aware that there is actually no time of year when you're safe from ticks. We are still collecting adult black-legged ticks here outside this week. Last week, every week we've ever been out when there's not snow on the ground, we've been able to at least collect some ticks. And so just making people aware that yes, summer is the high tick season. And we don't have as many black-legged ticks down here in the Southeast as we do up north, but you are really always at risk. So always do your tick checks when you come back inside. So let's talk a little bit about two different ticks and two different models that we've done based on these data that we've collected. So the first one is the black-legged tick, Ixodes scapularis. It is the most common, most known tick, I should say. It's not as common in the Southeast. The northern variant is a little more aggressive. It's moved down through Roanoke area of Virginia. It's now into North Carolina in the Research Triangle, and we're not sure how far south it's going to go. But it is a known vector of Lyme disease. So this is the Lyme disease related tick. It's of concern considerably around the country. It also can transmit the pathogen that causes anaplasmosis and babesiosis and Powassan virus and a whole bunch of other things. So this is not one you want to encounter, preferably, but it is also very a stealth little guy and tiny. So I was interested in working with this a little bit, and then, as I mentioned, I started doing modeling in grad school in the 90s, and there was very few mathematical models of tick-borne diseases back then. And one of the very few that was out there was this program called Limestone. It was created as a life history model by some folks at the USDA, and they were interested in understanding the kind of dynamics I'm talking about. And so they took some mosquito life history models and turned them into a tick life history model. And then they started out with a whole bunch of different processes. They had a cattle tick model, and then they made the Lone Star model, and then they had a dog tick model. And they finally, as Lyme disease came around in the 90s, they converted it to a black-legged tick model, and then they made Limestone. And as Lyme disease became more of an issue, they decided to make this into a user-friendly DOS application. In my little grad school life, I wrote a nice letter to these faculty at the USDA, so Gary Mount and Daniel Hale. And Mount was in the middle of retiring, but he took a moment to write me a letter back and included a floppy disk that had Limestone on it. So fast forward to 2015 timeframe, and I get a call from some folks at the CDC who are interested in trying to revisit Limestone, because honestly, since Limestone came out, there's really not been any modeling done for black-legged ticks or Lyme disease. And they were really interested in bringing this Limestone back to life and updating it with what we know, but they couldn't find anybody who had a working copy of it or knew how to recreate one. So I dig through my files and pull out the floppy disk, and then find a friend who has a floppy disk reader, and then install a DOS box on my MacBook. And surprisingly, I was able to get mine to run and pull it back up. And so I ended up working with the CDC to indeed update this and to make it Limestone 2.0. So you never know when you have old floppy disks that they could come in handy. I've learned a lot about the program itself. I knew the papers as well that were based on it, and there are a lot of things that are really inconsistent mathematically as well as computationally in this model. That they made some cheats. I only have the executable. I would love to have the old code to see how they made it things stabilize in this system that should not stabilize if you look at the mathematics. But what we did instead was we really focused on creating this. This is an R program now rather than Visual Basic. It's still a vector host disease model. So I have black-legged ticks as my vector, and then I have broken out all of the different host types based on the importance of the ticks' use of them as blood meals and their vector capacities, their ability to be part of this disease cycle. And so if you know anything about Lyme disease or read about it, the white-footed mouse always comes up because it is an incredibly good food source for immature black-legged ticks, and it's also really good amplifying host, a vector reservoir competent host for the pathogen that causes Lyme disease. The model also had small mammals and birds, which are just kind of other things that the immatures might feed on that are reasonably good reservoir hosts, but not as good as white-footed mice. They have reptiles. Reptiles are actually refractory to the pathogen, and so they are good hosts, but they are not good for the pathogen. There's medium-sized mammals, which the adults will feed on, as well as the immatures. And there's white-tailed deer, which are wonderful tick food, but they actually are also refractory. They will not amplify. They don't get affected by Borrelia burgdorferi, the pathogen. So they are the main food source for the adults. And then adults will also feed on medium-sized mammals, and then the small immatures feed on these smaller animals. The original model had livestock. Again, it was based out of a cattle fever tick model. And livestock don't really play a role with Lyme disease, but what do are what I would term insectivores or shrews, which are small mammals that are feed on insects rather than grain. And it was important to put these in because a lot of the host-targeted methods we're going to talk about here in a second put out grain to attract the animals to it, and insectivores will not be attracted by that, but they are as good at a reservoir host as the white-footed mice. And so it's important in the model to have animals that will not be attracted by food, but that could play a role in the pathogen dynamics. So a little bit about the model itself. It's a matrix life history model. And so we walk through this on a weekly time step. So if I zoom in a little bit here, everything is weeks, right? So eggs get laid. They after a certain number of degree days, we have the cumulative degree weeks concept where we are keeping track of how many degrees above a threshold it is and adding that up over time. And once I can integrate to a point where I have enough warm days, the eggs will hatch. And once they hatch, they harden into larvae. The larvae then have a period of time in which they can find a host before they die, which is a maximum of 80 weeks. But what we found in the model is that if there's no penalty for questing, they stick around forever and ever. And this didn't seem to be logical in what we see in the field or what we see in the lab when we've reared them. And so what we did was we started giving a penalty of a fat burn that they use up their calories if they're questing. And so if they sit perfectly in diapause, ticks will do this when there's not the right conditions, when the weather's not warm enough, when it's too dry, when there's not long enough day length, they'll just go into diapause and they'll conserve their energy. But if the conditions are right, they'll either be walking around, they'll be up and active, and they'll be burning through their calorie reserves. And so we took a penalty of a 3-week questing penalty for every week they spend questing. And so that just shortens the maximum time that they can stay in that line. And then if they're lucky, they find a host and they feed on that host for a week. They fall off, they go through another molting period until it's warm enough and enough degree days. And then they'll emerge as the nymph, and then they repeat that process for nymphs, and then they repeat the process for for adults. And so and then cycles back, they lay their eggs, and we go back around again. One comment about this host seeking concept: when I wanted to be able to compare my data to my field data, when I go out and collect ticks, as I said earlier, especially with the 2020 data, it would seem that not every host seeking tick is available. And there are 2 ways that the host seeking ticks are not available. One is they're in that diapause. And so what I should consider host seeking, I'm now actually looking at questing. So I call these the surplus ticks, or the ones that will be up actually questing that did not find a host. So I assume the hosts get up earlier than I do and get there first. And so we only get the ones that are left. And so this is always a challenge with models, is that you know way too much, and how you scale that back to what you would actually collect in the field is a really interesting challenge. And so we came up with this solution that if we at least know what proportion are up questing, and then we let the hosts pick up first, and then whatever's left over is what I would be assumed I could find if I was out flagging. The host dynamics are really simple. We're just doing a birth death replacement with a constant population rate of one, the inverse of their lifespan. There is a maximum number of ticks for each life stage by host. We assume there's just there's a physical capacity of the surface area of an animal. And so while they're usually not covered head to toe in ticks, there are some data points, some data out there to give some estimate of how many larvae and nymphs feed on a host, and some adults as well. We're still working on trying to get better estimates, for example, of how many larvae will feed on a deer at a time. But we have some of the best guesses that are out there and put those into the model. The pathogen dynamics are vector-borne only. The original one had some transovarial model vector. So we assume that a infected tick will infect a susceptible animal that's reservoir competent, and that animal will be ill for a certain length of time and can infect the next susceptible tick that feeds on it. So vector-borne transmission. The original model had it where if an infected female laid eggs, a certain portion of them would be infected, but that's more likely because the testing they had back then didn't differentiate true Lyme disease pathogens from some of the other related pathogens. As I mentioned, only 4 of the host types are reservoir competent, with white-footed mice and insectivores being the most likely to transmit the pathogen to a tick, and small mammals and medium-sized mammals as being about half the competence of the first 2. So we put all these things in, and then we want to measure two things. And in the Lyme literature, the most common measurements they have are what's called DIN and DON. So I will measure sometimes the density of all nymphs for DON and measure the density of infected nymphs for DIN. And again, I'm measuring only the surplus of questing nymphs. The nymphs are the ones that are chosen because they're the least likely to be seen and the most likely to bite humans, particularly in Northeast and the Midwest of the United States. So we wanted to then validate the model. So we needed data to validate our model. So I pulled data from my data set here in Virginia, got some data from Carey Institute up in New York, and we got some data from some folks in Minnesota. And the weather, the only inputs the model takes is weather inputs. And so we took weather from U.S. National Weather Service at these stations closest to each of the field sites. And it uses daytime temperature, total precipitation, day length, and then an estimate of humidity out of that, or vapor pressure. And so we could collect those data on a weekly average basis for each of the 3 different sites and feed them into the model to see what we would get as results coming back out. So everything else in the model was kept consistent between the 3 different locations. And so what we could find if we ran this on Virginia, weather inputs, this is model output. And so the way you read these graphs is the year is along the bottom. So we pulled weather data for 2007 to 2016. And then this is an estimate of the number of ticks per 100 meters squared you would collect if you were out flagging. The black line is the density of all nymphs, and the red line is density of infected nymphs. And so what we can see from this is that we get back at a maximum of 15, but there's some variation from year to year between 5 and 15 here in Virginia. And we compared this to field data, and it was a fairly reasonable match for at least if you at least do a rank ordering statistic between the two. And so there's some variation, but you see that there's just like one major peak here in Virginia, fairly tight time frame, which is not untrue of what we find in the field. If I run the same exact model using the New York data, we could see that we get almost twice as many ticks in some years, up to 30 ticks per 100 meters squared. And there is some variation year to year, but it's a totally different pattern of which years were high and which years were low. And so again, in a much larger peak time, it's not the single peak that we get here in Virginia, very tight. And then there's a little tiny blips as well, usually in the late summer, predicted by the model, which again is not uncommon to be found out in the field in New York. If you look at Minnesota, on the other hand, it's kind of a much more wild and crazy collection, with some years having almost no nymph surplus, and some years having almost New York level 25 ticks per meter squared. And again, you can also see that the time frame in which they're out is actually broader, which is in some ways counterintuitive to what you would think. But the time frame is spread out a little bit more, because in Minnesota they actually end up overlapping into the following year, which has also been started to be found in the field. And the field data again in Minnesota is highly variable, and the ranks of when the year is the highest year in 2011 is what they found in the field data as well. And so the model seemed to reflect a lot of the key components of what we really wanted to understand about Lyme disease. And so here's the actual data lined up with field data. So the green are the field data, and the black lines are the model results that I was just talking about, right? And so we can see that at least for Minnesota and Virginia, we get very similar green lines. New York was a little bit harder to figure out, but what we ended up having to do was to varying the host dynamics are playing a bigger role, and so we have a lot more work to do still in New York. So the whole point of this model was actually to look at tick control. And so a little bit about how we can then apply the model now that we're a little bit confident of what we're doing. And so just to make sure that those who, again, don't work in this field have an idea of what we're talking about when we talk about tick control, is there's 2 major types. You can go out and you can spray chemicals or remove leaf litter, but most of the time people are spraying chemicals. And the biggest challenge with spraying the chemicals, which do kill the ticks that are up questing, is that it's very harmful for other invertebrates. So people sometimes will then say, well, what if we do host targeted controls? And so you can put out tick tubes and bait boxes that allow a white-footed mouse to treat themselves or other small mammals that are attracted to the bait that you put out, which is usually, again, grain. You put out grain, you put out some cotton that has permethrin on it. The animals pick up the cotton, take it back. And then in their nests, they're then treated for all the ticks as well as all the other ectoparasites. 4-posters are a method of doing the same kind of self-application for white-tailed deer. And so The challenge, of course, is it can lead to resistance to that chemical in the ticks. But more importantly, in our area, is more likely to alter the ecology for wildlife. So anytime you start feeding wildlife, you run into some potential problems. And so neither of these are ideal. But the question was, well, can we use them in some minimized manner that will actually give us some good results? And so this is what ITM is about, is, if I combine these to minimize the side effects and maximize tick reduction, what would be the benefits? But again, ticks live multiple years. And so in order to implement this in the field, it's very challenging, very expensive, very time consuming. We have those year-to-year variations where things could swamp out just because one year might be a low year or a high year. How do I know if that was really from the integration from the control I did, or was that just from from weather? So models are a great place to explore this options very cheaply and quickly. So we put these out, looking at these different treatments. So whether deer treatment, just put mortality in any tick that gets on a deer dies. We could have white-footed mouse treatment, so that additional mortality for any ticks feeding on the mice. And then the broadcast acaricide, the insecticide that we put out, we spray and we assume everything that quests for the next 4 weeks dies. And so if we just do deer treatment alone, these graphs are based on a consistent average weather input. And the little C's down here are the years that we treat. So you treat, it takes a couple of years to kick in, and then you get a couple of years of relief. But as soon as you stop, within 3 or 4 years, you're right back to where you were before. And you've dropped it by a quarter, but you're not really knocking it out. White-footed mouse treatment, you get a much quicker input and drop down, but it certainly only lasts for a year after treatment. And this is when you're treating 75, both of these are 75% of the hosts are treated, which is a lot of work. By broadcast acaricide, sure enough, it works really well. Remember, these are only killing the questing ticks, and so everything else that's in diapause or quiescent or in hibernation is not affected. And as soon as I stop, it comes back. And even more alarmingly, it comes back with a rebound effect. And so you actually get a larger number of ticks the following year after stopping this kind of treatment, which is of concern. So if we combine them, right, we said, well, maybe if we combine them, and we tried combining them in lots of different ways, and you could indeed get a little bit longer of an effect in the model, but you get this large rebound effect still, and it's extremely unlikely that you would be able to get 75% of any area treated at this level, and that's what it took to even get this kind of reduction. And so for this part of the model, we were definitely looking at we can make a difference. It takes lots of work and lots of years, and that the combination will eliminate it for years after the treatment. But of course, there's limitations here, right? We're in a closed system. It's spatially explicit. I'm assuming every animal is treated equally, which is not likely. And then everything is immediate. And the biggest thing is that there's these concerns about negative side effects of the controls that I have a lot of concerns about. And so what we've done in my lab here, I'm going to talk about for the last few minutes, is trying to look at a different control measure that is kind of flipping the whole idea of controlling ticks on their head. Because the whole goal with this kind of model is really looking at and understanding, can we eliminate ticks from the environment? I mean, people hate ticks. I go to a place and I'm like, I'm here to collect ticks. They're like, take them all. And it's just this hatred of them that makes us want to eliminate them. But there's concern about the ecological dynamics of removing any part of an ecosystem. And so what we've kind of done is we flipped it on its head and said, rather than thinking about eliminating ticks from the world, what if I eliminate ticks from my world? And so kind of flipping it more, thinking about when I come in from outside, I'm of course going to bring in some dirt. If I bring in dirt, the way I treat it is I have a vacuum that cleans it up. And so the TickBot was a semi-automatic control system that we worked on with some engineers to test. And it's a little self-driven car that drags a flag behind it. And that rather than spraying the chemical in the environment, we spray it on the flag, and the flag is dragged through the environment. So there's minimal residue left, but every tick that comes in contact with it will die. And so this was the idea that if I run this in my world, I should be able to control the ticks that are around me. Another theory that's out there quite often that I hear that we tested as well was the idea that we can do some kind of natural tick prevention. And so people often say, well, I'll get guinea fowl or I'll get chickens and they'll take care of the ticks in my backyard. And there's a couple papers. There's one that actually kind of looked at the use of guinea fowl for the black-legged tick, right? There's another student paper that looked at guinea fowl and the black-legged tick, but both of those were not really well done. And they certainly didn't look at the Lone Star tick, which is actually what I'm going to look at. And so there's this whole, how we avoid these nasty Lone Star ticks. So we know that these ticks will feed on some animals, but there's this concept that if I just put chickens out or turkeys or guinea fowl out, they'll eat the ticks. But no one had ever really looked at testing that. And so what we did is we went out and I had a grad student work on exploring whether or not that was true. And so again, our idea here was, I want my house in a very pretty area to live in a tick-free area that I can spend time in my yard, or maybe I can make this along a trail in a wooded area, and then let the ticks have their world outside of my world. And so we could run the tick bot along the edge, and I can put guinea fowl and let them run all over the property. And so the question was, can we look at how we could understand whether those can work? Do they work better together? And the ecological question for the guinea fowl on top of this was, there are no studies that demonstrate that the ticks don't feed on the guinea fowl. And so maybe having guinea fowl there will actually make more ticks, because unlike the tick bot, which can't feed them, even if the guinea fowl kill them, they could also be a food source for them, making it even more complicated ecologically. And so what we did was, in this particular case, my grad student built an agent-based model. And so looking at interactions at the small level, because I only have a few guinea fowl and I have one tick bot and I'm in a finite area, we took a lot of that matrix model approach, but looked at it at an individual tick, individual host model. And so that allowed us to look at these kind of variable parameter values. I could assign different habitats, different treatment regimes. I could understand which ticks were where in the environment to know whether they were affected by the tick bot or potentially able to jump on a guinea fowl or be able to be eaten by a guinea fowl. So it gave me all the structure that I needed, much more fine detail than the weekly based matrix model I just showed you. And this model is actually based on the Lone Star tick, right? So before we were talking about the black-legged tick, this is the one that has that we've collected tens of thousands of here in our area. So we've used all of those thousands of data points to be able to estimate the parameters for this model. This is the tick that's associated with the red meat allergy. And so we're interested in knowing what is going on with these, where they're spreading, and how we could possibly think about controlling something that we collect tens of thousands of a year. They're very aggressive and will bite humans. And so what we did was we go out and did a lot of field studies with the tick bot to be able to understand how many they killed. We did all kinds of different trials. We looked at trials in the lab to understand how far from the tick bot the ticks will be coming in from and what their kill rate would be at different distances away, so that we could put that into our model. If the tick bot runs in its own little zone, we knew that we would assume that we could kill and attract from a certain different distance away that we could actually put into our model again. We ran implement studies all over the place to be able to understand what this did and how often it killed. And we did mark recapture studies to put ticks out and see how many were killed. Right. So we did the same thing with guinea fowl. My grad students went up to a farm where they actually had guinea fowl, and they did enclosures and exclosures and in Gladstone, and they looked to see, well, if you put ticks in with the guinea fowl in one place and keep the guinea fowl out of another place and have an open pen, and they could find that they would do a lot of they'd go out, they'd collect the ticks, they'd paint them, they'd put them in, so we knew how many were in each spot. And they put the guinea fowl out for a couple hours, so they actually had the data. And what they found was in the black or the exclosure, so if no hosts are allowed in at all, the ticks just don't get picked up. And so there's an amazing number of recaptures. So it does speak to the fact that the animals come through, do pick up ticks. And so the open areas and the enclosures where the guinea fowl were not statistically different. And so it's not clear that they are actually doing much. They may have a little bit of a reduction, but I mean, overall, open and enclosures had about the same number of ticks recaptured. And so again, we put ticks in there to be able to recapture them. The other question I threw out there was whether or not guinea fowls were actually hosts. And so they actually collected all of the droppings off the guinea fowl at night where they roosted. And they actually, as you can see there, if you zoom in, did find engorged ticks falling off these. And so they are indeed picking them up, but probably not eating them, probably more likely being hosts for them. So we built a mathematical model, again, an agent-based model that allowed us to do all of the things. as I mentioned before, are basically the same kind of tick model. The hosts now include guinea fowl. And then we have this tick bot rather than a aericide spray. And then we would look to see how this all affects each other. And so again, we built the model with looking at the lawn. We had a tick bot, and we had a forest edge, and we had the forest, and we let the guinea fowl wander wherever they wanted to wander. And so we looked at all different options. My students looked at, whether there's control, how many guinea fowl you have, how many guinea fowl with different, with the tick bot, without the tick bot, how many things you can measure. This is always a big challenge in any kind of model like this, is you get more data than you could possibly ever think about. And so if we look at the results, if we look how many ticks got killed by a tick bot with birds, right? So if there's no tick bot with the tick bot, you basically get slightly fewer ticks actually killed with the birds, because they are actually probably being absorbed by the birds. But tick bot itself will kill quite a few ticks running along the edge, right? And so she also measured how many questing ticks were eaten probably by the guinea fowl, assuming they eat a certain percentage of them, and maybe 83. So not nearly as effective as the tick bot itself. So finishing up here quickly, the reduction that you can get if you have both guinea fowl and tick bot in your yard is about 43%, which is not the 70-some% we looked at a second ago, but it's still a lot better than it's better for the environment perhaps that you want to do. And definitely without a tick bot, you only get about an 18% reduction, and probably most of those are actually absorbed, that would end up creating more ticks in the future. So conclusions, we need to still do it's very complicated, right? Especially once you have a potential agent that can feed back on itself. And we need a lot more studies, a lot more work on understanding how these systems all play off against each other, and what the optimal is, if there is an optimal. So certainly a lot of acknowledgement. The tick bot and the guinea fowl work is the work of Alexis White, who is now an entomologist in Long Island. But lots of other grad students and undergrads have worked on this, as well as my fellow faculty. And shout out to the funding for the NIH and CDC and the EPA for a lot of this work. With that, I will take questions in the time I have left.
Great, thank you so much, Holly. I really enjoyed your presentation. It's nice to see mathematicians get out into the field and to be able to really create cross-disciplinary research. We do have a few minutes, so we'll take one from Carolyn Williams. Her question was, you mentioned the importance of time off host to manage ticks, for instance, impacting environmental humidity by removing Japanese barberry in New England. I don't see an additional question to that, but maybe you could expand a little bit about the value and how that time off of host is important for managing ticks.
Yeah, I mean, I think off time, the kind of environmental change is interesting in changing the local dynamics of the humidity. I mean, ticks are incredibly, what's the right word? Tough, right? I mean, they're really hard to kill. So while the Japanese barberry, I think, also affects hosts moving in, getting the humidity change at the ground level can be very, very difficult to a significant enough level to make a difference, because it's at least in our area here, we're so close to sea level that you're not going to affect the humidity at the ground. And so it is an interesting thought of what they can and can't do off host. But we've tried putting ticks underwater to see how long they live, because we thought maybe we could flood them out. And at 70 some days, we still had them alive. So they really don't drown. They don't, you could desiccate them, as said. I mean, the environmental humidity is a big deal, but in at least with sea level rise and everything around here, you're not going to get humidity low enough, I don't think.
Yeah. And we have another one from Benki Olatosi. He said, can your model be made specific to residential areas versus uninhabited areas? Second, would population density be an important input to consider?
So certainly, as you saw with the last bit of the model there, I mean, we have some that the Limestone model is typically thought of as a little bit more open area. And then we moved to the agent-based approach. We're also looking at making our Limestone spatially explicit, so that we can then look at populated areas and unpopulated areas and how that would impact all the different potential host movements and all that. So there's a lot I'm working with some folks up in New England as well to really look at using agent-based models in very specific layouts of the environment to understand how things move through. And so, yeah, it is very important, and just adds more complication, which would be the next step after we understand what we are doing.
Well, thank you, Holly. If anyone has any additional questions, you can go ahead and type them into the chat and she can answer later. But we will go ahead and close this session out and remind you guys to take a 10-minute break and to come back at 10:30. But thank you so much, Holly, for your time today. We really enjoyed it.
Thank you.